So average size of human gene is 3000 bases. DNA fingerprinting is what? It is a comparison of DNA from different sources to establish the identity. What is VNTR? Variable number of tandem repeats. So those are, it is also called RFLP. Hello dear students, welcome back to session 9 of this chapter called Molecular Basis of Inheritance. Myself, B. Shobharani from the Department of Biology, Vidhyaashram Pre-University College, the Temple of Excellence. By seeing this picture, I think definitely you would get to know that yes, in today's session, I'm going to talk about the fingerprinting, that is DNA fingerprinting. Before I move on to DNA fingerprinting, the continuing part of Human Genome Project, what are the outcomes of Human Genome Projects? So what will be the outcomes of the Human Genome Project? So the first draft of Human Genome Project was announced on 26th June 2000 and the project was completed in April 2003. What are the important results that have been obtained through this Human Genome Project? First, very important, total number of functional genes in human cells are about 30,000, much lower than the expected number of about 1 lakh. First very important outcome of Human Genome Project is what? Total number of functional genes in human cells are about 30,000, much lower than the expected number of 1 lakh. Second very important outcome is the human genome has 3,164.7 million nucleotide base pairs. So what is the number of base pairs? A human genome has 3,164.7 million nucleotide bases. That means it forms about how much? 99.9% of bases are exactly the same in all the people. So there is only a difference of 1%. So 99.9% bases are exactly the same in all the people. Only 2% of the DNA actually code for proteins. The other 98% of DNA functions what is known as the junk unknown DNA function. It is called junk DNA. So only the 2% of the DNA molecule actually codes for proteins. So remaining sequence of DNA, we call it as an unknown DNA or a DNA with unknown function called junk DNA. So which is called as non-coding sequence. We can also use the word called VNTR. That is, I'm going to explain that in the coming session. So what are the outcomes of HGP? Here you can see the first draft of Human Genome Project was announced on 26th June 2000 and the project was completed in April 2003. The important results obtained were what? Total number of functional genes in human cells are about 30,000, much lower than the expected number of about 1 lakh. And the human genome has what? 3,164.7 million nucleotide pairs and 99.9% of the bases are exactly same in all the people. So the difference lies in only 1% of the base pairs. Then only 2% of the DNA actually code for proteins. The other 98% of DNA functions is unknown, and that this DNA with unknown function is called the junk DNA. Average size of human genes is 3,000 bases. Next, very important, this is usually they'll ask in exam. What are the outcomes of Human Genome Project? So average size of human gene is 3,000 bases. The largest human gene is dystrophin gene with 2.4 million bases. So average size of human gene is 3,000 bases. So the largest known gene is what? Of dystrophin gene with 2.4 million bases. Chromosome 1 has maximum number of genes, and why? that is chromosome X has maximum number of genes, that is 2968, and Y chromosome has the lowest number of genes, that is 231. So as you know, there are two types of chromosome X and chromosome Y. Chromosome X has maximum number of genes and Y chromosome has the lowest number of genes. The functions of about 50% of the discovered genes are unknown. See, though we say DNA is the main genetic material which is very much responsible for the passing on of the characters, but with the Human Genome Project, with the study, the functions of about only 50% of the discovered genes is known and the 50% is unknown. Scientists have identified about 1.4 million locations where single base DNA differences that is occur in human. This information promises to revolutionize the processes of finding chromosomal locations for diseases associated sequences. So these are all the outcomes of Human Genome Project. Now, what are the applications of HGP? Here you can see there are wide applications of HGP, and here you can see that these are all the outcomes of the HGP. As I already said, chromosome 1 has 2968 gene, 2% gene codes for protein, total genes 30,000, average gene consists of 3,000 bases, then 3164.7 million nucleotides, functions of 50% discovered genes is unknown, repetitive sequences contribute large portion, and largest gene is the dystrophin gene. So what are the applications of Human Genome Project? It helps in complete understanding of human biology and genetics. Very, very important. So the concepts or the outcomes of Human Genome Project will help to understand the human biology and genetics. Then many genes associated with human diseases like cystic fibrosis, breast cancer, muscular disorders, cardiovascular disorders, diabetes, blindness, etc. are identified. So by this, we can identify a good number of diseases that are caused in human beings. So this helps in understanding the disease development and formulating new methods of diagnosis and treatment. So all this will help to come out with better techniques of identifying the disease and with a better technology and methodologies of diagnosing the disease, because as we know that diagnosing the disease is very, very important for proper medication. Only when the disease is diagnosed that this is the correct disease, this is the reason for the disease, then only it will be very, very easy for the treatment. So this Human Genome Project will definitely help to overcome all these problems. So it catalyzed the development of biotechnology industry, particularly use of information technology for information. Like we have this bioinformatics, where we apply the computing techniques or computing methodologies to store the huge human data. Storage and development of gene-based therapeutics. So even as we know that science and technology, particularly in the field of medicine, it has advanced so much that even the treatment methodology can be improved to a greater extent to overcome all these diseases. So these are all the applications of HGP. Even this is very important from examination point of view, where they'll ask what are the applications of HGP. So it helps in complete understanding of human biology and genetics. Many genes associated with human diseases like cystic fibrosis, breast cancer, muscular disorder, cardiovascular diabetes, blindness can be identified. And this helps in what? Understanding the disease development, formulating new methods of diagnosis and treatment. Then it is also used in many technology, like it might be particularly in the use of information technology for information storage and development of gene-based therapeutics. Moving on to next very important concept called DNA fingerprinting. Here you can see the picture itself will tell you what is DNA fingerprinting. So DNA fingerprinting is what? Comparison of DNA. DNA fingerprinting is what? It is a comparison of DNA from different sources to establish the identity. And the DNA fingerprinting technology has been applied to wide aspects to overcome many problems. Like it might be very important to identify the culprits who have committed the crime. So DNA fingerprinting is nothing but it is comparison of DNA from different sources to establish the identity. So this is a technique which helps in establishment of identity of a person, detecting and comparing specific nucleotide sequences in the DNA of the individual. So when we compare, when we do the comparison of the DNA sequence from one individual to the other individual, then we can arrive at a conclusion whether that particular person has committed the crime or not. So this DNA fingerprinting is particularly very important in the study of, in the particular field called forensic science. So this is a technique which helps in the establishment of identity of a person by detecting and comparing specific nucleotide sequences in the DNA of individuals. So this technique was first time developed by Alec Jeffrey from Leicester University, UK. What is the principle behind this DNA fingerprinting? For example, like there are many cases where the crime has been committed, but later on the culprit is held responsible for the crime. So this is only possible only when they go for this DNA fingerprinting, and even it can also overcome the problems like disputed parentage. So when they claim for the parentage also that time, so this is a one very important technique which is going to come to the rescue of that particular parents who have this disputed parentage. So each individual has a unique sequence of DNA. So what is the principle behind this DNA? How are they going to analyze? By comparing the DNA sequences you are taking from different sources. So each individual has a unique sequence of DNA. So you have a different DNA sequence, I have a different DNA, the other one has a different DNA sequence. And certain segments of DNA have base sequences repeated many times. Right. The length of these sequences and the number of times they are repeated in the DNA is unique to a person. This is the principle behind DNA fingerprinting. So how do we do the comparison? The length of these sequences, DNA sequences means what? Base pairing sequences, and the number of times they are repeated in the DNA is unique to a person. So it differs from a person to person. The length of the sequences and how many times the sequences are repeated, it differs from a person to person. So by analyzing the length of the sequence of the DNA and the number of times it is repeated, so it is possible to identify the particular, for example, like somebody has committed one crime, and that can also be solved by doing this comparative study of the DNA sequence. So this is the principle that is held behind this DNA fingerprinting. So what is DNA fingerprinting? As I said, it is a method of comparing the DNA sequences taken from different sources. And this is a technique which helps in the establishment of identity of a person by detecting and comparing specific nucleotide sequences in the DNA of individuals. So this technique, as I said, it was developed by Alec Jeffrey from Leicester University, UK. And what is the principle that is held behind this DNA fingerprinting? Each individual has a different unique sequence, and the length of the sequence and the number of times the sequence is repeated varies from a person to person. So based on this, they are going to analyze the DNA fingerprinting. Now, VNTRs. So DNA fragments, that is variable number of tandem repeats. So this is asked for 1 mark question also usually. So expand VNTR. What is VNTR? Variable number of tandem repeats. So those are, it is also called RFLP. It is called RFLP, that is restriction fragment length polymorphism. So DNA fragments with repeating sequences are called variable number of tandem repeats. So what is VNTR? DNA fragments with repeating sequences are called variable number of tandem repeats or restriction fragment length polymorphism, which varies from a person to person. Then what is DNA polymer? Inheritable mutation observed in population at high frequency is called DNA polymorphism. So inheritable mutation observed in a population at high frequency is called DNA polymorphism. Now, what are the steps of DNA fingerprinting? Here you can see the steps so here the sample is taken, then it is RNA is extracted, it is taken for gel electrophoresis, then northern blotting. So the labeled parts, then we can go for this X-ray film. So these are all the steps of DNA fingerprinting. Now let us study one by one. The steps involved in the DNA fingerprinting technique is, first one is what? Collection of the sample. First, very, very important is first the sample, DNA sample should be collected. So what are the sources of the DNA samples? It might be blood, or it might be even the semen. For example, if a rape has been taken place, then the semen also is very important. Then the hair, then the saliva or any other tissue even in the dried state are collected. So those are the sources of DNA content. So first and foremost is collection of the sample, maybe from the site of crime or from the individuals who have overcome that crime. So even the victims also, from the victims it is collected. So samples such as blood, semen, hair, saliva or any other tissue even in the dried state are collected. Next, extraction of DNA. So the material, the DNA extract, whatever is collected from the sources is subjected to high speed centrifugation. So the collected material is subjected to high centrifugation. So this will separate the DNA from the cell. So this centrifugation process will help to separate the DNA. So here you can see the sample is taken, then it is separated. Next, fragmenting DNA. So with this, then the DNA in the sample is treated with restriction endonucleases. As you know, they are called as molecular scissors, which is going to cut the DNA molecule into fragments, smaller restriction fragments. So what is the first step in DNA fingerprinting? Collection of the sample. Next step is extraction of the DNA from the sample by subjecting it to centrifugation. Then fragmenting the DNA. That is, the DNA in the sample is subjected to a restriction endonuclease to obtain smaller restriction fragments. Next, separating DNA fragments. Next step, the fragments of DNA are made to run on agarose gel plate by electrophoresis technique, in which, depending on molecular size and shape, the nucleotides produce distinct bands in gels. So next is what? Separating DNA fragments. The fragments of DNA are made to run on an agarose gel by electrophoresis technique. Depending upon the molecular size and shape, the nucleotides produce distinct bands in the gel. Next, extracting DNA from the gel. Now, the double-stranded DNA fragment in the gel is denatured into single strands by treating the gel with alkaline solution. Then by Southern blotting, very, very important technique in DNA fingerprinting, through Southern blotting technique, these single-stranded DNA fragments are transferred onto nitrocellulose sheet, or it is also called as the nylon membrane, for visual observation. So then by observing the bands, then we are able to analyze the DNA sequence in that particular sheet. So first is what? Separating DNA fragment. The fragments of DNA are made to run on agarose gel plate by electrophoresis technique, in which, depending on molecular size and shape, the nucleotides produce distinct bands in gel. Then extracting DNA from gel. So the double-stranded DNA fragment in the gel is denatured by single strands by treating the gel with alkaline solution. Then by Southern blotting technique, these single strands, that is the DNA fragments, are transferred onto a nitrocellulose sheet or a nylon membrane for visual observations. Then tagging radioactive probes. Probe is what? It is nothing but a radioactive labeled synthetic DNA. The nitrocellulose sheet is now incubated within these radioactive probes. The probes act as nucleotide detectors and binds to specific nucleotides present in DNA fragments on the sheet. So next is what? Tagging the DNA fragments to radioactive probes. Probe is what? As I said, it is a radioactive labeled synthetic DNA. The nitrocellulose sheet is now incubated with these radioactive probes. The probes act as what? Specific nucleotide detector. So it is going to detect that particular sequence of DNA on the nitrocellulose sheet and present in DNA fragments on the sheet. Then autoradiography. The next step is what? The nitrocellulose sheet is X-ray photographed. Only such DNA bands to which radioactive probes bind appear as dark spots in the X-ray photographs, which is called as autoradiograph. Very, very important. Based on that bands on that nitrocellulose sheet, then the comparison is done between the two DNA samples that has been collected from two different sources. So on visual or computer analysis, two genetic fingerprints can be compared to know the degree of similarities and difference. So the very important concept of DNA fingerprinting is on the comparison, whether it is similar or dissimilar. So based on that concept only, they will arrive at the right decision of claiming who has committed the crime. So these are all the important steps in DNA fingerprinting. Now, what are the applications of DNA fingerprinting? DNA fingerprinting has wide applications. As you know, this is one of the very important technique that comes to the rescue of many of victims, particularly like the one who have, even particularly some culprit has committed the murder, or even some culprit has committed the rape, or even for the disputed parentage, or for all that, I think the answer is DNA fingerprinting. So it is used in the field of medicine, environmental microbiology, then food and agriculture, as I said, even in forensic science and phylogenetic. That is what you can call it as the relationship. So identification of criminals in cases connected with rape, murder, that is in forensic investigation. In solving paternity disputes to identify the real father of the children. So this is a very common thing which you come across, particularly in maternity homes. When the babies are born, they look alike. So sometimes there are chances of the exchange of the babies. So in that case, if they claim for their child, and if it is not possible to arrive at a correct decision, then they can go for fingerprinting, where they can arrive at the correct decision of claiming the parentage of the child. Then to identify the victims of disasters such as fire, floods, landslides, and vehicle accidents. Sometimes, the bodies will be, will be beyond recognition. So they will be in such a state that maybe fire victims or maybe landslides, when they come across that even vehicular accidents where the whole body is crushed. So in that case, it is very difficult to identify the person. So in that case, even it might be some natural disasters, like even the floods and everything, the whole body will be decomposed. So in that case, so it is very difficult to arrive at who that person is. So in that case, also the DNA fingerprinting will help to claim the identity. Then changes in the gene, that is mutation and its effect, also can be studied. So as I said, it has wide applications for the welfare of mankind, like it can be used in the identification of criminals in cases connected with rape, murder, etc., particularly in forensic science, you can say. Then in solving paternity disputes to identify the real father of the children, and to identify the victims of disasters such as fire, floods, landslide, vehicle accidents, where the body is beyond recognition and it is so very decomposed, in a decomposed state or dismantled state, that it is very difficult to identify the person. So in that case also, DNA fingerprinting is going to help. And changes in the gene, that is, if at all any change in the gene has, suppose a gene has undergone mutation, and as I said, it may lead to many harmful problems. So in that also it can be studied. Hope you have understood all the concepts what I have explained in this wonderful chapter called Molecular Basis of Inheritance. So this is a very important chapter from examination point of view. So I'll be back in the coming session with a very new chapter called chapter 14, that is ecosystem. So till then, goodbye and thank you.